Okay, class, today we're going to talk about translation in Chapter 15, nucleic acids. This far, we've already talked about nucleotides and the difference between DNA and RNA and how base pairing occurs and how DNA can be replicated to form more DNA in a semi-conservative process to give copies of DNA to daughter cells so that they can also make proteins and you can replicate cells in living organisms. And also how you can transcribe a DNA molecule using the template strand in order to form mRNA or messenger RNA. That mRNA then can be translated into proteins, and that's what we're going to cover today, translation. So there are different RNA molecules I've previously mentioned in other videos. mRNA, messenger RNA, is RNA synthesized from the DNA in transcription. It carries genetic information from nucleus to cytoplasm in order to make proteins, so nucleus being an organelle and the cytoplasm kind of being the space in between organelles within a cell. mRNA contains codons, which is a three-letter sequence, three nucleotides that code for a specific amino acid. We will talk more about what a codon is and how tRNA, in fact, helps build proteins using that codon. tRNA, transfer RNA, carries correct amino acid that correspond to specific codon on mRNA. So they have to match in order for tRNA to bring the amino acid and then form peptide bonds and then send out a protein. It contains an anticodon, which is a three-nucleotide sequence that's complementary to the codon on messenger RNA in order for it to bind to mRNA. Then rRNA, ribosomal RNA, is an RNA molecule that forms ribosomes where translation occurs. So you can kind of think of ribosomes like a workbench. As a blacksmith hammers out a product, so the ribosome gets a protein hammered out of it. So RNA to protein, this is translation. It's the final part of our central dogma. It's the process in which the information encoded in mRNA is used to direct the sequencing of amino acids to synthesize a protein. So here's how it works. Here's mRNA. You just got this from your DNA being transcribed. Remember, you should see no thymines and you should see uracils, and it's single-stranded and runs from the 5' to the 3' end. Well, as it turns out, if you're coding for a protein, that comes from a gene in DNA, and mRNA is composed of three-letter sequences which are called codons. And so AUG is a codon, ACG is a codon, GAG, so on and so forth. And each codon corresponds to a certain amino acid in protein synthesis. So AUG corresponds to the amino acid methionine. ACG corresponds to the amino acid threonine, so on and so forth. And so that is how information from DNA gets transcribed to RNA and how those three-letter sequences build proteins, okay? And so the question is, how does this work? And it uses tRNA. Okay, tRNA, transfer RNA. This is tRNA right here. And here is your messenger RNA with its three-letter sequences called codons. And so as you see, AUG right here is methionine. And so what happens is tRNA comes and finds the AUG. And the way it binds to AUG in order to transfer methionine to start building amino acids is tRNA has an anticodon. So the anticodon is going to be the complementary sequence to the codon on mRNA, which means in order for it to bind to A, U, and G, it must be U, A, and C. Okay, which remember how the base pairing works. And so UAC must be it, and it's going to be ran in the opposite direction, 3' to 5'. And the entire tRNA molecule contains a methionine for this particular one. So as you can imagine, there's a bunch of different tRNAs, one for every single amino acid. And for every amino acid, it also contains an anticodon to place that amino acid in the correct position, thus building you a nice protein. You do not have to memorize this table, but you will need to know how to use this chart in order to solve problems, I think, on sapling. So let's just delete one of those so that we can practice reading about codons, okay? So I'm going to get a little colorful here. So this it's asking you to build the protein corresponding to this segment of messenger RNA. And so first we need to find AUG. Okay. Well, AUG, the way you read this, is the first base, and you're going to look for A. And so here's A. And then we need the second base right here, which is U. So we're going to be looking somewhere in this portion. And then it says it's a G for the third base. And so we're going to be looking right here. Let me do some deleting. I just wanted to show you how this chart works. So this is exactly where we're going to be looking at. AUG, it's the start codon. It's going to be methionine, okay? So we should do an MET for methionine. It's the three-letter abbreviation for that amino acid. Then UUC, we're going to be looking right here, and then find C as your third base right here. UUC is going to be phenylalanine, so it's going to be PHE. And then UUC, or sorry, we just did UUC. It's going to be AAA, so A, then A. It's going to give you tyrosine. No, that's lysine, excuse me. So now we have done that one, and finally we need GAG. So that just puts us right here. There's A and then G. So we got glutamic acid, okay, GLU. So hopefully that gave us the right one. And you can do number 2 on your own. Try doing it without looking at the answer, maybe by blocking out the answer as I just did right there, okay? But that's all you have to be able to do, okay? So if ever you're asked to do this on a test, this chart would be given. And so that's how mRNA is used to build proteins. You guys can finish up the chapter on your own. I hope that this was valuable and helpful information. Bye-bye.